Tonight's lecture is over the history of life. So we'll start with a quick review of some of the stuff we were talking about in the last lecture. Descent with modification. In natural selection, Darwin argued that this was the outcome of three principles that operated in nature. One was that most characteristics of organisms are inherited or passed down from parent to offspring. Secondly, more offspring are produced than are able to survive, so resources for survival and reproduction are limited. Thus, there is a competition for those resources in each generation. Thirdly, offspring vary among each other in regard to their characteristics and those variations that are inherited. Darwin and Wallace both thought that offspring with inherited characteristics that help them compete will survive and have more offspring than those individuals with variations that are less able to compete. Because characteristics are inherited, these traits will be better represented in the next generation. This will lead to a change in populations over generations in a process that Darwin described as descent with modification. Furthermore, natural selection leads to greater adaptation of the population to its environment as the beaks of the finches, which are adapted to different food sources. And we can see drawings of those finches on your slide. The driving force behind evolution is natural selection. Natural selection can only work if variation exists among organisms. Variation arises ultimately from genetic mutations. Diversity is further encouraged through sexual reproduction. As environments change, selective pressures shift and favor different adaptations. In this way, given thousands or millions of years, species evolve. Moving on, when two species evolve in diverse directions from a common point, it is called divergent evolution. Such divergent evolution can be seen in the forms of the reproductive organs of flowering plants which share the same basic anatomies, but look very different as a result of adaptation to different kinds of pollinators. And the pictures on your slide show examples of that. In other cases, similar phenotypes evolve independently in distantly related species. For example, flight has evolved in both bats and insects, and they both have structures we refer to as wings. Wings are adaptations to flight. However, the wings of bats and insects have evolved from very different original structures. This phenomenon is called convergent evolution, where similar traits evolve independently in species that do not share a recent common ancestor. Charles Darwin gathered important evidence for his theories during an around-the-world voyage. He was particularly struck by the diversity of animals on the Galapagos Islands off the coast of Ecuador. Darwin regarded adaptation to the environment and the origin of new species as closely related processes. If some geographic barrier, say an ocean separating islands, isolated two populations of a single species from each other, the populations could diverge more and more in appearance as each adapted to local environmental conditions. Over many generations, the two populations could become dissimilar enough to be designated as separate species. Darwin realized that explaining such adaptation was the key to understanding descent with modification or evolution. This focus on adaptation helped Darwin envision his concept of natural selection as the mechanism of evolution. Darwin synthesized the theory of natural selection from two observations that by themselves were neither profound nor original. Others had the pieces of the puzzle, but Darwin saw how they fit together. Darwin based his mechanism of natural selection on two undeniable facts and an inescapable conclusion. One observation Darwin made was that any population of a species has the potential to produce far more offspring than the environment can possibly support with available resources such as food and shelter. This overproduction leads to competition among the varying individuals of a population for these limited resources. Another observation that Darwin made was that individuals in a population of any species vary in many inherited traits. No two individuals in a population are exactly alike. You know this variation to be true of human populations. Careful observers find variation in populations of all species. So we can draw a conclusion from those observations, and that is, in the struggle for existence, those individuals with traits best suited to the local environment will, on average, have the greatest reproductive success. They will leave the greatest number of surviving, fertile offspring. Therefore, the very traits that enhance survival and reproductive success will disproportionately be represented in succeeding generations of any given population. It is the unequal reproductive success that Darwin called natural selection, and the product of natural selection is adaptation, that is, the accumulation of favorable variations in a population over time. So let's discuss an example of this. Polar bears and grizzly bears, as closely related as they are, each display an evolutionary adaptation, that is, white fur and brown fur respectively. And that resulted from natural selection operating in their respective environments. Presumably, natural selection tended to favor the fur color that provided each bear lineage with the best camouflage in their home territories. As the process leading to adaptation, natural selection is also the mechanism of evolution. Moving on, Darwin found convincing evidence for the power of unequal reproduction in examples of artificial selection, that is, the selective breeding of domesticated plants and animals by humans. We humans have been modifying other species for millennia by selecting breeding stock with certain traits. The plants we grow for food bear little resemblance to their wild ancestors. This is because humans have customized crop plants through many generations of artificial selection by selecting different parts of the plant to accentuate food. Moving on, the power of selective breeding is especially apparent in our pets. Pets have been bred for fancy and for utility. For example, people in different cultures have customized hundreds of dog breeds as different Basset Hounds and St. Bernards, all descended from wolves. The tremendous variety of modern dogs reflects thousands of years of artificial selection by humans. So in artificial selection, humans substitute for the environment in the screening of heritable traits of populations. Moving on, species is a Latin word meaning kind or appearance. Indeed, we learn to distinguish between the kinds of plants and animals, between dogs and cats, for example, or between roses and dandelions, from differences in their appearance. Although the basic idea of a species as distinct life forms seems intuitive, devising a more formal definition is not so easy. One way of defining a species is the biological species concept. It defines a species as a group of populations whose members have the potential to interbreed with one another in nature to produce fertile offspring, that is, offspring that can reproduce. Geography and culture may conspire to keep a Manhattan businesswoman and a European dairy farmer apart, but if the two did meet and mate, they could have viable babies that develop into fertile adults because all humans belong to the same species. In contrast, humans and chimpanzees, despite having a shared evolutionary history, remain distinct species because they do not interbreed. We cannot apply the biological species concept to all situations. For example, basing the definition of species on reproductive compatibility excludes organisms that only reproduce asexually, that is, producing offsprings from a single parent. Fossils aren't doing any sexual reproduction either, so they cannot be evaluated by the biological species concept. In response to such challenges, biologists have developed several other ways to define species. For example, most of the 1.8 million species named to date have been classified based on measurable physical traits. Another approach identifies species in terms of ecological niches, focusing on unique adaptations to particular roles in a biological community. Yet another classification scheme defines a species as the smallest group of individuals sharing a common ancestor and forming one branch on the tree of life. Each species concept is useful depending on the situation and the questions being asked. The biological species concept, however, is particularly useful when focusing on how discrete groups of organisms may arise and be maintained by reproductive isolation. Moving on, clearly, a fly will not mate with a frog or a fern, but what prevents closely related species from interbreeding? What, for example, maintains a species boundary between the eastern meadowlark and the western meadowlark? Their geographic ranges overlap in the Great Plains region, and they are so similar that only experts can tell them apart, and yet these two bird species do not interbreed. A reproductive barrier is anything that prevents individuals of closely related species from interbreeding. Let's examine the different kinds of reproductive barriers that isolate gene pools of species. We classify reproductive barriers as either prezygotic or postzygotic, depending on whether they block interbreeding before or after the formation of zygotes, that is, fertilized eggs. Prezygotic barriers prevent mating or fertilization between species. The barrier may be time-based, that is, temporal isolation. For example, western spotted skunks breed in the fall, but the eastern species breeds in the late winter. Temporal isolation keeps the species from mating even when they coexist on the Great Plains. Another example of a prezygotic barrier is habitat isolation. In this case, species live in the same region but not in the same habitats. For example, one species of North American garter snakes lives mainly in the water, while closely related species lives on the land. Traits that enable individuals to recognize potential mates, such as a particular odor, coloration, or courtship ritual, can also function as reproductive barriers. This is called behavioral isolation. In many bird species, for example, courtship behavior is so elaborate that individuals are unlikely to mistake a bird of a different species as one of their own kind. In other cases, the egg-producing and sperm-producing structures of different species are anatomically incompatible. This is called mechanical isolation. Also, gametes, that is, the egg and sperm of different species, are incompatible. This prevents fertilization, and this is called gametic isolation. Gametic isolation is very important when fertilization is external. Male and female sea urchins of many species release eggs and sperm into the sea, but fertilization only occurs if species-specific molecules on the surface of egg and sperm attach to one another. On the other hand, postzygotic barriers operate if interspecies mating actually occurs and results in hybrid zygotes. In this context, hybrid means that the egg comes from one species and the sperm comes from another species. In some cases, hybrid offspring die before reaching reproductive maturity. That is a reduced hybrid viability. For example, although certain closely related salamander species will hybridize, the offspring fail to develop normally because the genetic incompatibilities between the two species creates a non-viable offspring. In other cases of hybridization, offspring may become vigorous adults but are infertile. That is, they have a reduced hybrid fertility. A mule, for example, is the hybrid offspring of a female horse and a male donkey. Because mules are sterile, there is no avenue for gene transfer between the two parental species, the horse and the donkey. In other cases, the first-generation hybrids are viable and fertile, but when these hybrids mate with one another or with either parent species, the offspring are feeble or sterile. This is called hybrid breakdown. For example, different species of cotton plants can produce fertile hybrids, but the offspring of the hybrids may not survive. In summary, reproductive barriers form the boundaries around closely related species. In most cases, it is not a single reproductive barrier, but some combination of two or more that keeps species isolated. Moving on, a key event in the origin of many species occurs when a population is somehow severed from other populations of the parent species. With its gene pool isolated, the splinter population can follow its own evolutionary course. Changes in its allele frequencies caused by genetic drift and natural selection will not be diluted by alleles entering from other populations. An example of that is gene flow. Such reproductive isolation can result from two general scenarios. One is called allopatric speciation, and the other is called sympatric speciation. Moving on, as one species changes over time, it branches to form more than one new species, repeatedly, as long as the population survives or until the organism goes extinct. Moving on, several geological processes can fragment a population into two or more isolated populations. A mountain range may emerge and gradually split a population of organisms that can inhabit only lowlands. Or a land bridge, such as the Isthmus of Panama, may form and separate the marine life on either side. Or a large lake may subside and form several smaller lakes, with their populations now isolated. Allopatric speciation can also occur if individuals colonize a new geographically remote area and become isolated from the parent population. An example is the speciation that occurred on the Galapagos Islands following the initial colonization by immigrant organisms. Moving on, how formidable must a geographic barrier be to keep allopatric populations apart? The answer depends partly on the ability of the organism to move about. Birds, mountain lions, and coyotes can cross mountain ranges, rivers, and canyons. Nor do such barriers hinder the wind-blown pollen of pine trees or the spread of seeds carried by animals. In contrast, small rodents may find a deep canyon or a wide river an impassable barrier. Allopatric speciation is more common for a small, isolated population because it is more likely than a large population to have its gene pool changed substantially by both genetic drift and natural selection. For example, less than 2 million years ago, the few foreign animals and plants that first colonized the Galapagos Islands gave rise to all the species now found there. But for each small, isolated population that becomes a new species, many more simply perish in their new environment. Life on the frontier is harsh, and most pioneer populations become extinct. Even if a small, isolated population survives, it does not necessarily evolve into a new species. The population may adapt to its local environment and begin to look very different from the ancestral population, but that doesn't necessarily make it a new species. Speciation occurs with the evolution of reproductive barriers between the isolated population and its parent population. In other words, if speciation occurs during geographic separation, the new species cannot breed with its ancestral population, even if the two populations should come back into contact with one another at a later time. In sympatric speciation, a new species emerges from an old one, even though they live in the same time in the same place. So how can a subpopulation become reproductively isolated while in the midst of its parent population? In some cases, subgroups of populations evolve adaptations for exploiting food sources in different habitats, such as the shallow versus deep habitats of a lake. In other cases, a type of sexual selection in which females choose mates based on color can also contribute to rapid reproductive isolation. But the most frequently observed mechanism of sympatric speciation involves large-scale genetic changes that occur in a single generation. Biologists have documented many examples of plant species that originated from accidents during cell division. Such accidents can produce organisms with extra sets of chromosomes. A new species that evolves this way has polyploid cells, meaning that each cell has more than 2 sets of chromosomes. The new species cannot produce fertile hybrids with its parent species. This is a good example of sympatric speciation because reproductive isolation has occurred in a single generation without geographic isolation. And in this next slide, we see one form of sympatric speciation that can begin with a series of chromosomal errors during cell division. In a normal cell division event, chromosomes replicate, pair up, and then separate so that each new cell has the same number of chromosomes. However, sometimes the pairs separate and the end cell product has too many or too few individual chromosomes in a condition called aneuploidy. And in this next slide, we see that in some cases, a polyploid individual will have 2 or more complete sets of chromosomes from its own species in a condition called autoploidy. The prefix auto means self, so the term means multiple chromosomes from one's own species. Polyploidy results from an error in meiosis in which all of the chromosomes move into one cell instead of separating. So what does this have to do with speciation? Well, these new gametes will be incompatible with the normal gametes produced by this particular plant species. However, they could either self-pollinate or reproduce with other autoploidy plants with gametes having the same diploid number. In this way, sympatric speciation can occur quickly by forming offspring with a 4N genome, which is called a tetraploid. These individuals would immediately be able to reproduce only with those of this new kind and not those of the ancestral species. Moving on, another form of polyploidy occurs when individuals of 2 different species reproduce to form a viable offspring called an allopolyploid. The prefix allo means other. Remember allopatric speciation? Therefore, an allopolyploid occurs when gametes from 2 different species combined. And the figure on this slide illustrates one possible way an allopolyploid can form. Notice how it takes 2 generations or reproductive acts before the viable fertile hybrid can result. The cultivated forms of wheat, cotton, and tobacco plants are all allopolyploids. Although polyploidy occurs occasionally in animals, it takes place most commonly in plants. Animals with any of the types of chromosomal aberrations described here are unlikely to survive and produce normal offspring. Moving on, scientists have discovered more than half of all plant species studied relate back to a species evolved through polyploidy. With such a high rate of polyploidy in plants, some scientists hypothesize that this mechanism takes place more as an adaptation than as an error. Moving on, what is the tempo of speciation, or what is the rate of speciation? Biologists continue to make field observations and devise experiments to study evolution in progress. However, much of the evidence of evolution comes from the fossil record, that is, the chronicle of extinct organisms engraved in layers of rock over millions of years of geologic time. What does this record say about the process of speciation? In this cartoon on this slide, we see a model called the gradual model, wherein species that are descended from a common ancestor diverge gradually in form as they acquire unique adaptations. And that brings us to the punctuated equilibrium model. Many fossil species appear suddenly in a layer of rock and persist essentially unchanged through several layers or strata until disappearing as suddenly as they appeared. The term punctuated equilibria describes these long periods of little change or equilibrium punctuated by abrupt episodes of speciation. So the cartoons on this slide and the previous slide show two different models for the tempo of speciation, that is, the gradual model and the punctuated model. In the punctuated model, there are no transitional stages in the lineages. The new species change little, if at all, once they appear. In the gradual model, differences evolve little by little in populations as the species become adapted to their local environments, and new species evolve gradually from the ancestral population. According to the graduated model, big changes or speciations occur by the steady accumulation of many small changes. But what do the punctuated and gradual patterns tell us about how long it takes a new species to form? Suppose that a species survived for 5 million years, but most of the changes in its body features occurred during the first 50,000 years of its existence. Just 1% of its overall history. Time periods this short often cannot be distinguished in the fossil strata. Thus, based on its fossils, the species would seem to have appeared suddenly and continued with little change before becoming extinct. Even though such a species may have originated more slowly than its fossils suggest, a punctuated pattern indicates that speciation occurred relatively rapidly. For species whose fossils show much more gradual change, we cannot tell exactly when a new biological species formed. It is likely that speciation in such groups occurred relatively slowly, perhaps taking millions of years. How often do species evolve abruptly and then remain essentially unchanged for most of their existence? Rapid speciation is certainly known to occur. As we saw earlier, abrupt speciation can occur by polyploidy in plants and even occasionally in a few animals. And genetic drift and natural selection can significantly alter the gene pool of a small, isolated population in just a few hundred generations. Regardless of the tempo of speciation, as species diverge, differences accumulate and become more pronounced, eventually leading to the formation of new groups of organisms that differ greatly from their ancestors. Furthermore, as one group produces many new species, another group may lose species to extinction. The cumulative effects of multiple speciation and extinction events have helped to shape the dramatic changes documented in the fossil record. This is a type of macroevolution. Moving on, the fossil record, which is the sequence in which fossils appear in the rock strata, is an archive of macroevolution. Sedimentary rocks are the richest resources of fossils and provide a record of life on Earth in their strata. The fossils in each stratum of sedimentary rock are a local sample of the organisms that existed at that time in history. By studying many sites over the past 2 centuries, geologists have established a geologic timescale. This reflects a consistent sequence of geologic periods. The timeline presented here on your slide is separated into 4 broad divisions: the Precambrian, which is a general term for the time before about 540 million years ago, followed by the Paleozoic, Mesozoic, and Cenozoic eras. Each of these divisions represents a distinct age in the history of Earth and its life. The boundaries between eras are marked by mass extinctions when many forms of life disappeared from the fossil record and were replaced by species that diversified from the survivors. For example, the beginning of the Cambrian period is delineated by a great diversity of fossilized animals that are absent in the rocks of the late Precambrian. And most of the animals that lived during the late Precambrian became extinct at the end of that era. The fossil record reveals that the evolutionary history of life on Earth has been episodic, with long, relatively stable periods punctuated by brief, cataclysmic ones. During these upheavals, new species formed and others died out in great numbers. Extinctions are inevitable in a changing world, but the fossil record reveals a few instances of great change, times when the majority of life on Earth, between 50 and 90% of all living species, suddenly died out, disappearing forever. Scientists have documented 5 such mass extinctions during the last 500 million years. Today, many biologists believe that we are witnessing a 6th mass extinction. Human activities are modifying the global environment to such an extent that many species are currently threatened with extinction. In the past 400 years, and that is a very short time on the geological timescale, more than 1,000 species are known to have become extinct. Scientists estimate that this is 100 to 1,000 times the extinction rate seen in most of the fossil record. Do these events represent the beginning of a mass extinction? This question is difficult to answer, partly because it is hard to document both the total number of species on Earth, estimates range from 10 million to 200 million, and the number of extinctions. Although losses have not reached the levels of the other quote-unquote big 5 extinctions, the populations of many species are declining at an alarming rate. This suggests that a 6th human-caused mass extinction could occur within the next few centuries. And, as with prior mass extinctions, life on Earth may take millions of years to recover. But the fossil record also shows a creative side to the destruction. Mass extinctions can pave the way for the evolution of many diverse species from a common ancestor. Fossils are a reliable chronological record only if we can determine their ages. The record of the rocks reveals only the relative ages of fossils and therefore the order in which groups of species evolved. However, the series of sedimentary layers alone does not tell us an absolute age of the embedded fossils. The rock strata are analogous to the layers of wallpaper that you might peel from the walls of a very old house. You could determine the sequence in which the wallpapers had been applied, but not the year in which the layer was added. Geologists use a variety of methods to determine the ages of rocks and the fossils they contain. The most common method is called radiometric dating, which is based on the decay of radioactive isotopes. Recall that isotopes are alternate forms of elements. The dates in the geologic time scale in the figure on your slide were established by radiometric dating. Moving on, when most people think of evolution, they envision a much more dramatic transformation. But how can we account for such evolutionary products such as flight in birds and braininess in humans? Birds are derived from a lineage of earthbound reptiles. How could flying animals evolve from flightless ancestors? More generally, how do major novelties of biological structure and function evolve? One way evolutionary novelty arises is when structures that originally played one role gradually acquire a different role. Structures that evolve in one context but become adapted for another function are called exaptations. This term suggests that a structure can be adapted to alternative functions. It does not mean that a structure somehow evolves in anticipation for future use. New features can arise gradually via intermediate stages, each functioning in the organism's current context. Exaptation can account for the evolution of novel structures. Consider the evolution of birds from a dinosaur ancestor. Birds have lightweight skeletons with honeycombed bones, a feature found in their dinosaur ancestors. Since the fossil record clearly indicates that light bones predated flight, they must have had some function on the ground. The ancestors of birds were small, agile, bipedal dinosaurs that would have benefited from a light frame. Moreover, a wing-like form with feathers would have increased the surface area of the forelimbs. These enlarged forelimbs were adapted for flight after functioning in some other capacity such as thermal regulation, courtship displays, or camouflage, and those are roles that wings still play today. The first flights may have been only glides or extended hops as the animal pursued prey or fled from a predator. As flight evolved, animals with better adapted feathers and wings would have had a selective advantage. The concept that biological novelties can evolve by the remodeling of old structures is in the Darwinian tradition of large changes being an accumulation of many small changes crafted by natural selection. Moving on, gradual evolutionary remodeling, such as the accumulation of flight adaptations in birds, probably involves a large number of genetic changes in populations. In other cases of macroevolution, just a few genetic changes can become magnified into major structural differences between organisms. Such cases are of particular interest to scientists working in the field of evo-devo, or evolutionary developmental biology, which studies the evolution of developmental processes in multicellular organisms. Genes that program development control the rate, timing, and spatial pattern of changes in an organism's form as it develops from a zygote into an adult. A subtle change in a species' developmental program can have profound effects. The same or very similar genes are involved in the development of form across multiple lineages. Changes in the number of nucleotide sequence and regulation of these genes have led to the huge diversity in body forms. Many striking evolutionary transformations are the result of a change in the rate or timing of developmental events. For example, a salamander called an axolotl illustrates a phenomenon called pedomorphosis, which is the retention into adulthood of features that were solely juvenile in the ancestral species. The axolotl grows to full size and reproduces without losing its external gills, a juvenile feature in most species of salamanders. Pedomorphosis has also been important in human evolution. Humans and chimpanzees are even more alike in body form as fetuses than they are as adults. In the fetuses of both species, the skulls are rounded and the jaws are small, making the face rather flat. As development proceeds, uneven bone growth makes the chimpanzee skull sharply angular, with heavy brow ridges and massive jaws. The adult chimpanzee has a much greater jaw strength than we have, and its teeth are proportionately larger. In contrast, the adult human has a skull with decidedly rounded, more fetus-like contours. Put another way, our skull is pedomorphic. It retains fetal features even after we mature. Our large skull is one of our most distinctive features. Our large, complex brain, which fills the bulbous of the skull, is another. The human brain is proportionately larger than the chimpanzee brain because growth of the organ is switched off much later in the human development. Compared with the brain of a chimpanzee, our brain continues to grow for several more years, which can be interpreted as a prolonging of a juvenile process. Homeotic genes, the master control genes we described in a previous chapter, determine such basic features as where a pair of wings or legs will develop on a fruit fly. Changes in such genes or in where such genes are expressed can profoundly affect the body form. Consider, for example, the evolution of tetrapods, that is, animals with backbones and 4 limbs from fishes. The location within a developing limb where certain homeotic genes are expressed is initially the same in both fishes and tetrapods. A second region of expression in the developing tetrapod limb, however, produces the extraskeletal elements that develop into foot bones. Thus, changes in the expressions of these genes appear to have led to the evolution of walking legs from the paired fins of fishes. Duplications of homeotic genes probably facilitated the origin of new body shapes in animals, including the evolution of vertebrates, that is, animals with backbones, from invertebrates, animals without backbones. For example, consider the homeotic genes of a fruit fly, an invertebrate, and a mouse, a vertebrate. A fruit fly has a single cluster of homeotic genes that direct the development of major body parts, whereas the mouse has 4 clusters of these genes. 2 duplications of these gene clusters appear to have occurred in the evolution of vertebrates from invertebrates. Mutations in these duplicated genes may then have led to the origin of novel vertebrate characteristics. For example, some genes may have taken on new roles, such as directing the development of a backbone, jaws, and limbs. Moving on, the phylogenetic tree of life, which Darwin first used, is the classic tree of life model describing phylogenetic relationships among species, and it is the most common model that scientists use today. New ideas about horizontal gene transfer and genome fusion have caused some to suggest revising the model to resemble webs or rings. Horizontal gene transfer, also known as lateral gene transfer, is the transfer of genes between unrelated species. Horizontal gene transfer has been shown to be an ever-present phenomenon, with many evolutionists postulating a major role for this process in evolution, thus complicating the simple tree model. Genes have been shown to be passed between species which are distantly related using standard phylogeny, thus adding a layer of complexity to the understanding of phylogenetic relationships. And the table on this slide summarizes the mechanisms of prokaryotic and eukaryotic horizontal gene transfer. I will let you spend a little bit of your own time going over this table. Moving on, recall from previous chapters that we've talked about the endosymbiotic theory before. Scientists believe that the ultimate horizontal gene transfer occurs through genome fusion between different species of prokaryotes when 2 symbiotic organisms become endosymbiotic. This occurs when one species is taken inside of the cytoplasm of another, which ultimately results in a genome consisting of genes from both the endosymbiont and the host. The mechanism is an aspect of the endosymbiote theory, which is accepted by a majority of biologists as the mechanism whereby eukaryotic cells obtain their mitochondria and chloroplasts. Moving on, in Doolittle's phylogenetic model, eukaryotes evolved from many species sharing genes by horizontal gene transfer. It is hypothesized that individual prokaryotes from different species transferred the bacteria that caused mitochondrial development and chloroplastic development in eukaryotes and archaea. And in this slide, we see the ring of life model, which, according to the ring of life phylogenetic model, the 3 domains of life evolved from a pool of primitive prokaryotes. So let's recap tonight's lecture. You should be able to describe the theory of evolution and how it works. You should be able to define a species, and you should know how a new species can form. You should also be aware of how populations evolve. And you should be able to read and analyze phylogenetic trees that document evolutionary relationships, and that ties in to the previous chapter.